Welcome to the Deep Dive, the shortcut to fluency in the most exciting new science, where we take complex research and give you the knowledge you need fast.
Today, we're wrestling with a problem of just massive scales, one of the biggest roadblocks in modern functional genomics. I mean, we want to systematically map these huge, complex regulatory circuits that, govern how our cells behave.
Right, especially in really critical systems like our immune cells. We want to ask a simple question. If we tweak gene A, what happens to the other, 20,000 genes?
Exactly. And the go-to technique for that has been this thing called Perturb-seq.
Perturb-seq. So that's pooled CRISPR screens with a single-cell RNA sequencing readout. It's been revolutionary, really. It lets you go in, knock out specific genes, and then see the exact consequences at super high resolution inside individual cells.
It's a brilliant tool. Absolutely brilliant. But it has a pretty fatal flaw, at least in its conventional form.
Which is?
It just doesn't scale. I mean, if you're working with precious cells, say from a patient, or you want to test thousands of genes, the cost just becomes prohibitive.
Right.
You're talking millions of dollars, billions of cells.
And it's not just a financial cost, right? When you try to study genetic interactions, like what happens when you change gene A and gene B together, that complexity, the number of cells you need, it just explodes exponentially.
That's the challenge. And our mission today is to analyze the source material, detailing a really profound solution to this. It's called compressed Perturb-seq, or CPS.
Compressed, okay.
Yeah, and it's a total game changer because it takes a technique straight out of electrical engineering, actually, compressed sensing theory, and it uses it to just fundamentally rewrite the rules of genetic screening.
So here's the core insight, the aha moment we want you to walk away with. This compressed approach, it gets you the same, gold standard accuracy as the old way.
But with efficiency gains that are just. They're measured in orders of magnitude. The papers report a tenfold, sometimes more, cost reduction.
It takes functional genomics from being this niche luxury to a tool you can actually use for high throughput discovery.
Exactly.
So let's start with the basics. Why does conventional Perturb-seq fail at scale? You said it combines CRISPR screens and single-cell RNA-seq.
Precisely. And don't get me wrong, it's given us incredible insights. I mean, we understand the innate immune response better. We've learned how autism risk genes function. Its power is in that resolution.
But the math just doesn't work out for large screens. Why does it hit that scaling wall so hard?
It's the definition of linear scaling. So if you want to test in perturbations, let's say you're knocking out different genes, you conventionally have to do roughly individual experiments.
One for one.
One for one. You test 500 genes. You need 500 distinct observations. You want to test the whole genome, 20,000 genes. You need 20,000 experiments. Every single gene you add, it adds linearly to the cell count, to the cost. It's unsustainable.
That sounds like a physical limit. I mean, how do you even get around that without just throwing information away?
See, you don't try to beat the physics. You exploit a biological truth, which is sparsity. Okay, tell us more about what that means in this context.
So if you knock out a gene, it doesn't cause some, chaotic, random change across the entire transcriptome. Regulatory systems are, they're sparse. Most of the time, a perturbation only affects a very small, select group of other genes, or maybe a co-regulated gene program.
So the vast majority of those 20,000 genes are just unchanged.
Exactly. The information we're collecting is mostly zeros. We're paying a huge amount of money to measure what isn't happening.
Okay, so the big realization is that if the output signal is inherently sparse, then you don't actually need to measure every single input individually. This sounds like the core idea of compressed sensing.
That's the leap. Compressed sensing, which, came out of signal processing, lets us perfectly recover a sparse signal from way, way fewer measurements than the old linear method requires. Okay, let's make this concrete. Say we're in a recording studio. Conventionally, we'd record 100 different singers, one by one, to hear what each of them sounds like.
All right.
With compressed sensing, what's the new plan?
Instead of 100 separate recordings, maybe you only do 5. But in each of those 5, you record a random combination of those 100 singers, all singing together. We call these composite samples.
Sounds like a mess.
It does. But you know a secret. You know that only a small handful of those singers are actually hitting the high notes. Those are your non-zero effects, your sparsity. So you can use some pretty sophisticated math to untangle that random noise and pinpoint exactly which singer was doing what. You can infer their individual voices perfectly.
That's amazing. And the gain in efficiency is moving from linear to logarithmic. Can you explain why logarithmic is such a huge deal here?
Logarithmic scaling is the dream. It means your cost grows so much slower than the number of things you're testing. You could double the size of your experiment from 500 genes to 1,000, and your cost. It might only go up by a small constant factor, not double.
So instead of needing n samples, you need something like k times log n.
Exactly, where n is the total number of genes, and k is that small number of genes that actually have an effect.
Yeah.
And because k is so tiny compared to n, you save measurements logarithmically.
But Perturb-seq is super high-dimensional. We're not just looking at one thing, like cell survival. We're looking at 20,000 gene expression levels at once. Does this logarithmic magic still work?
It does, but the math has to account for two really crucial biological complexities. It does that with two variables, r and q. The sample requirement scales with q plus r times log n.
Okay, let's define those from a biology perspective, not a math one. What is r?
So r is the rank. The easiest way to think of R is as the number of distinct sort of departments in the cell, these big co-regulated gene programs.
Okay.
So when a perturbation happens, the cell doesn't randomly change 20,000 things. It dials up the inflammation department or dials down the antiviral department. R is just the number of those core departments.
So R is the modularity of the output. What about Q?
Q is the complexity of the input. It's the maximum number of genes we knock out, the perturbations that have any real effect on any single one of those departments. Even if you knock out 500 genes, probably only a handful of them, that's a low Q, will actually touch the inflammation department.
So because biology is modular, effects are bundled into a small number of programs, R, and only a small number of inputs, Q, can disrupt them, the math holds up. Even if N, the total genes you test, gets huge, R and Q stay small. And that's what guarantees those incredible logarithmic savings.
That is the foundational insight. That's what makes all of this possible.
All right, quick recap. We've established that conventional Perturb-seq is basically crippled by linear scaling. Too expensive, too many cells. Compressed Perturb-seq, or CPS, breaks this limit by exploiting biological sparsity.
Right, the fact that most genetic effects are zero.
Which allows us to measure random composite samples instead of individual ones, and that delivers these logarithmic cost savings based on those two key numbers. The small number of core gene programs, R, and their relevant inputs, Q.
You got it.
So on to the lab work. How do you actually make these random composite samples? The source material lays out two different strategies.
The first one is called cell pooling. So normally in single-cell RNA-seq, you want exactly one cell per droplet. With cell pooling, you do the opposite. You deliberately overload the system. You pool cells, each with one unique genetic knockout, and you get multiple cells crammed into a single droplet.
So the gene expression profile you read out is the average effect of all the cells in that one droplet. You're physically averaging the signal.
Exactly. The second method is guide pooling. And this one is achieved by using a high multiplicity of infection, or MOI. Instead of one CRISPR guide RNA per cell, you infect a cell so it randomly receives multiple different guide RNAs.
Ah, so you get multiple gene knockouts happening simultaneously inside a single cell.
Correct.
That's a fascinating difference. Cell pooling mixes the effects between cells in a droplet, while guide pooling mixes the effects inside one single cell. What's the big advantage of guide pooling?
Guide pooling is really the only way, of the two, that you can estimate not just the first-order effects, what happens when you knock out gene A, but also those crucial, higher-order genetic interactions.
The non-additive stuff. What happens when you knock out gene A and gene B together?
Yes, and that is absolutely essential for mapping out the actual circuitry.
It sounds riskier, though. If you hit one cell with 3 or 4 perturbations at once, aren't you worried about cellular toxicity? The cell just dying and skewing your data?
That's the trade-off, absolutely. Guide pooling gives you the most information per cell, but you have to be very careful about cellular stress. Cell pooling is simpler, but you can get technical noise from the averaging, which might dilute some of the weaker signals.
Okay, so you've got your pooled samples. Now you need the computational muscle to untangle them.
And that engine is called FR-Perturb. It stands for Factorize Recover for Perturb-seq.
Okay, walk us through it. What are the steps that make this algorithm powerful enough to denoise this incredibly complex data?
It's a 3-step process. The first step is sparse factorization. It uses an algorithm called sparse principal component analysis on the gene expression matrix. Its only job is to find those few fundamental co-regulated gene programs we talked about, the R factors. It basically forces the data to reveal its underlying modular structure.
So it's a denoising step right up front. It focuses the entire analysis only on these big biologically meaningful blocks of signal.
Precisely. Step 2 is sparse recovery. Now they have the factors, we use a different sparsity-promoting algorithm called
A
lasso.
lasso. I've heard of that.
Right. It stands for least absolute shrinkage and selection operator. And we use it to learn how our perturbations affect those factors we just found. The power of lasso is that it's designed to mathematically force any effect that's small or noisy all the way to zero.
So it's exploiting that Q assumption we talked about, that most knockouts won't affect any given program. It just filters out all the noise.
It's the engine of the compressed part. It mathematically enforces the sparsity we already know exists in the biology. And the final step is just simple matrix multiplication. You combine the cleaned up effects on the factors with the gene weights from step 1, and you reconstruct the estimated effect size for every single one of your 20,000 genes.
So CPS is this whole package. It's new experimental methods to get information-dense samples combined with a custom computational engine, FR-Perturb, that's built from the ground up to leverage the sparsity of gene regulation.
That's it.
So let's see this in action. The researchers applied CPS to a really complex real-world problem, perturbing 598 immune-related genes in human macrophage cells. And they stimulated them with LPS, which is, a core model for inflammation.
Yeah, and they targeted a great mix of genes. The known LPS response genes were in there, but also hundreds of genes linked to diseases like IBD and susceptibility to infection. And the results, well, the results confirming the efficiency claims are just staggering.
Let's hear them. What about the cell pooling knockout screen first? How much did they save?
Okay, so they compared one single massively overloaded cell-pooled channel 1 experiment to 19 separate conventional channels.
1 versus 19.
And they found that cell pooling achieved a 4 to 20-fold cost reduction. They calculated they'd need 5 or 6 of the conventional channels to get the same validation accuracy and correlation in R value of 0.92 as they got from just one of their cell-pooled channels.
4 to 20 times cheaper for the same quality of data? That is the definition of a breakthrough. What about guide pooling, the method that gets you the genetic interactions?
Guide pooling, which they used with CRISPR interference for knockdown, showed similarly robust gains. By focusing on the cells that got 3 or more guides, they measured about a 10-fold reduction in the number of cells they needed, and so a 10-fold reduction in cost. And the effect size is still correlated really strongly with the conventional data, an R of 0.80.
That's still a very strong correlation.
It is. And it's important to point out that the FR-Perturb algorithm itself is a breakthrough. They showed it massively outperformed older methods, like ElasticNet, even when they applied it to the old conventional data. That factor analysis part just acts as a powerful universal denoiser for these sparse single-cell screens.
So after they combined all the data conventional, compressed, knockout, knockdown, they had this incredibly powerful data set. What new biology did this clarified map of the LPS response actually reveal?
The first thing is that the perturbations neatly organize the cell's response into clear structural components. They found 4 co-regulated programs P1 to P4 and 3 co-functional modules M1 to M3.
And what were they?
P1 was the classic inflammation program. Think cytokines, chemotaxis.
Yeah.
P3 was the antiviral program, the type I interferon response.
And did these programs behave as expected? Did it confirm what we already knew?
It did, which is a great sanity check. They saw a strong anti-correlation between P1 inflammation and P3, the antiviral response. And that confirms this known regulatory trade-off where cells generally don't go all in on both at the same time. It showed the map was biologically sound.
Okay, let's zoom in on the modules, the inputs. M1 was the core activators. Who was in that club?
So M1 had all the usual suspects, the heroes of the LPS response. MYD88, IRAC1, TREFX. As you'd expect, knocking those out just shut down the inflammation program. But M1 also had some novel players, including 2 genes involved in ER transport, RAB5C and PGM3.
Which points to a bigger role for protein trafficking and maybe N-glycosylation inflammatory signaling than we previously thought.
Exactly. And then you have M3, the module of inhibitors, the brakes on the system.
Right, so that included known suppressors like ZFP36. Knocking them out should cause inflammation to surge.
And it did. But here's one of the most compelling discoveries. M3 also identified 2 genes with no obvious immune function. XPR1, which is a retrovirus receptor, and Kittens 220, a scaffold protein. And their effects were almost perfectly correlated, an R value of 0.97.
That high a correlation suggests they're working together, maybe as part of a physical complex?
Precisely. And the experimental follow-up was beautiful. They confirmed their inhibitory role. Knocking out either XPR1 or Kittens 220 caused IL-6 secretion, a major inflammatory cytokine, to shoot up by about 2.6-fold. This is the power of CPS, finding these brand new regulators that are acting just as strongly as the core famous signaling molecules.
Now, for the advanced stuff, genetic interactions. We know guide pooling was designed for this. What did they find?
So the number of possible gene pairs is just astronomical, so the study didn't have the power to detect individual pairwise interactions. But what they did, which was very clever, was aggregate the perturbations into 490 functional modules. And by doing that, they did succeed in detecting significant intramodule interactions.
And what did CPS, with guide pooling, find that the much larger conventional screen missed?
This is a critical point. The guide pool data detected significant interaction effects in 3 specific biological modules, including regulation of chromosome organization and TNF-alpha signaling, that were completely invisible in the conventional dataset.
Wow. So it definitively shows the enhanced statistical power of the compressed approach for finding that kind of complex regulatory logic.
Without a doubt.
Okay, finally, let's connect this all back to human disease. They took these findings and integrated them with GWAS data, the genome-wide association studies.
Yeah, the idea was to see if these functional maps could point to disease-relevant genes. They created a signature for each perturbed gene based on its downstream targets, and then they asked a simple question. Is the genetic heritability for immune diseases enriched near those target genes?
And the answer
Was a resounding yes. They found 23 signatures linked back to 16 of the perturbed genes that showed significant heritability enrichment for traits like IBD, eczema, and rheumatoid arthritis.
And what's so valuable about that?
It's invaluable because this method successfully flagged those core activators like TRKF6 and MYD88. These are genes that are notoriously difficult to link to common diseases with a standard GWAS
Why is that?
because they're under strong negative selection. Evolution basically purges common, highly disruptive variants in these critical genes from the population. So GWAS, which relies on common variants, just can't see them.
So CPS gives you a functional causal map that lets you bypass that evolutionary blind spot of traditional GWAS.
Precisely. But this leads us to probably the most surprising and thought-provoking finding in the whole study.
The disconnect.
The disconnect. So when they compared the gene regulatory links they learned from their experiment to something called trans eQTLs, which are genetic regulatory links observed in real human populations, they expected to see a strong overlap.
But they didn't?
They didn't. In fact, they found that their experimentally derived links were not enriched for the population-level trans eQTLs. The correlation was actually slightly lower than random.
That's a huge discrepancy. I mean, how do you even begin to explain that? Are we measuring 2 totally different things?
We might be. I mean, one possibility is just a lack of statistical power in the current eQTL studies. But there could be fundamental biological differences between the immortalized cell line they used and primary human cells.
Or something deeper.
Or something deeper. The philosophical implication is that an acute engineered sledgehammer perturbation, a CRISPR knockout, might trigger a cellular response that is mechanistically distinct from the response to a subtle, lifelong genetic variation that exists in a natural population.
It forces you to ask a really tough question. Can we really use this incredibly powerful engineered perturbation map as a complete proxy for natural human variation?
It reminds us that we have this amazing tool for mapping causality, but we still have a huge challenge in reconciling that causality with real-world population genetics. This is where the field has to go next.
So, to sum it all up, compressed Perturb-seq, driven by cell and guide pooling and the FR-Perturb algorithm, fundamentally changes the economics of functional genomics. It moves it from this limited, resource-intensive tool to a truly scalable platform.
And that scalability unlocks 2 huge things. First, the cost savings for massive screens. And second, the enhanced power you need to see complex biology, like genetic interactions and those disease genes that are hiding from GWAs.
We noted that the most efficient guide pooling performance came from cells that had 4 or more guides. The researchers suggest this can be scaled up even further. So as you think about the future of mapping our genetic construction manual, here's the question for you to consider. How high can these efficiency gains ultimately scale before the biology of the cell itself just imposes an insurmountable barrier?
That's the billion-dollar question.
We'll see you next time on The Deep Dive.